Hello everyone. See, there are a number of methods for isolation of DNA from various kinds of samples, but the principle of DNA isolation is the same in all the methods, and the principle of DNA isolation is based upon precipitation of DNA from the aqueous solution. So today I am going to discuss the science of DNA precipitation, how DNA is actually precipitated out of the aqueous solution. So for precipitation of DNA, what do we add? We add two things. One is sodium salt and the other is ethanol or isopropyl alcohol. Both are required for precipitation of DNA from the aqueous solution. Now let us see how the sodium salt and ethanol carry out precipitation of DNA from the aqueous solution. So let us first have a look on the structure of DNA. So DNA is a highly polar molecule because it has a phosphodiester backbone which contains lots of phosphate groups. So DNA has lots of phosphate groups, and these phosphate groups give DNA a net negative charge. So DNA has a net negative charge and it is a highly polar molecule, and that is the reason DNA is highly soluble in water. So how DNA is gets dissolved in water, and how DNA remains soluble in water? DNA remains soluble in water by formation of hydration shell. Water forms hydration shell around the DNA molecule. So it is just like this. Okay, like this. A hydration shell is formed around the DNA molecule, and this keeps the DNA molecule dissolved in the water. Now what happens when the salt is added, when we add the salt into the aqueous solution of DNA? The moment we add salt, suppose we add sodium chloride. So when we add sodium chloride into the aqueous solution, the sodium chloride gets converted into, or gets like dissociated into, sodium ion and chloride ion. Okay. Now the moment we add sodium chloride into the aqueous solution, the sodium ions, they interfere with the hydration shell of the DNA molecule. Okay. Sodium ion interfere with the hydration shell of the DNA molecule by like forming bond with the phosphate group, because sodium is a positively charged ion and phosphate is negative. So sodium tries to form bond, tries to interact with the phosphate group. So sodium ion tries to form the electrostatic bond with the phosphate group, and thereby it interferes with the hydration shell of the DNA, and thereby it weakens it. Okay. When we add sodium chloride, when we add the sodium salt, sodium ion basically weakens up the hydration shell of the DNA molecule. Okay. So what happens? Now the sodium ion would form electrostatic bonds with the phosphate group of the DNA molecule, like this. Okay. So now what happens when you add sodium chloride or sodium salt into the aqueous solution? It is there, aqueous means water is there. Okay, water medium is there, water is a solvent here. So what happens in the water? The bond between phosphate and sodium ion is weaker. Okay, bond between phosphate and sodium ion is weaker. So how it is weaker? Now let me discuss this by looking at the Coulomb. What does Coulomb law say? Coulomb law says F is equal to 1 divided by 4 pi epsilon r into q1 q2 divided by r square. Okay, so Coulomb laws say that in a solution the force between the two ions Q1 and Q2, the two ions, oppositely charged ions, depends upon the dielectric constant of the solvent. Okay, ER is the dielectric constant of the solvent. In our case, Q1 is sodium ion and Q2 is the phosphate ion. Okay, so the dielectric constant of water is 80. Okay, so when we put 80 here, that means the force between sodium ion and potassium ion would be 80 times less stronger, right? So in water, because the dielectric constant of water is very high, because water is a highly polar solvent, okay, so the dielectric constant is very high, and dielectric constant actually tells about the ability of the solvent to keep the two oppositely charged ions away from each other. Okay, so it has a high dielectric constant, that means it has the ability to keep the sodium ion and potassium ion away from each other. So the bond between sodium ion and potassium ion is very weak in water. Okay, so by addition of only sodium salt, DNA won't be precipitated. Okay, by addition of sodium salt, only the hydration shell or the solubility of DNA is decreased in the water, but it is not completely, DNA don't get completely precipitated out in water. So for complete precipitation of DNA, we need to add ethanol. Okay, we need to add ethanol, and the dielectric constant of ethanol is 24. The dielectric constant of ethanol is 24, which is less as compared to water. That means in ethanol, the bond between the electrostatic interaction between sodium ion and potassium ion would be stronger as compared to water. Okay, so this is our DNA molecule. There are phosphate groups, and so the moment you add ethanol into it, since ethanol has a low dielectric constant, the bond between the potassium ion and the sodium ion becomes stronger, and that leads to disruption of hydration shell completely, and the DNA molecule pops out of the aqueous solution and it gets precipitated out. So this is the basic science behind DNA precipitation.